infections in addition to respiratory infection. Along with this, it can cause various pyogenic infection, genital infection, and non-suppurative complications such as acute rheumatic fever and post-streptococcal glomerulonephritis. So, regarding respiratory infection, sore throat, and acute rheumatic fever, which is a non-suppurative complication of group A streptococci, that we have studied in first year. So, in this second year, I will not be explaining in detail about Streptococcus pyogenes bacteria. Already you have studied it, but I will focus on skin infections only, which is caused by bacteria Streptococcus pyogenes. You just revise the chapter of Streptococcus pyogenes, which was from first year, and today I will focus on skin infections only caused by Streptococcus pyogenes. Among various skin infections caused by this bacteria Streptococcus pyogenes, there are 4 types of skin infections: impetigo, pyoderma, erysipelas, and cellulitis. What are the various skin infections caused by Streptococcus pyogenes? Impetigo, pyoderma, erysipelas, and cellulitis. What is impetigo? Impetigo means it is one type of skin infection caused by Streptococcus pyogenes, which is superficial, discrete. Discrete means separate. It is superficial, discrete, and crust spots will be seen mainly among children in case of impetigo caused by Streptococcus pyogenes. Impetigo, it is superficial, discrete, and crust spot mainly seen among children. It lasts for 1 to 2 weeks and it heals spontaneously without leaving any scar. Remember, SCAR, scar means घाउ सन्चो भएपछि कालो दाग बस्छ कि बस्दैन, that is called scar. Okay. Impetigo, it lasts for 1 to 2 weeks and it heals spontaneously without any scar. There is no scar formation. If there is healing of lesion, there will be no scar formation in case of impetigo. Next type of skin infection caused by Streptococcus pyogenes is pyoderma. One is impetigo, other is pyoderma. Pyoderma, it is more severe local skin lesion. Pyoderma is more severe local skin lesion with ulcerated area. There is ulceration in the skin, having raised and edematous edge. Lesion is raised and it is having edematous edge. It means lesion starts as vesicle. Vesicle means filled with clear yellowish fluid. Lesion starts as vesicle and later scar formation occurs over vesicle. Remember, there are 4 types of skin infections caused by Streptococcus pyogenes: impetigo, pyoderma, erysipelas, and cellulitis. In case of impetigo, there is no scar formation. In case of pyoderma, there is scar formation. Other type of skin infection caused by Streptococcus pyogenes is erysipelas. Literal meaning of erysipelas, it is Greek word and term erythros means red and pelas means skin. Red skin, erythros and pelas, combined it is called erysipelas. It is acute spreading lesion and infected area includes brawny edema with erythema. There is brawny edema. Brawny means having muscular strength. Strike ख्याल गरेको छ. There is no fluid-filled lesion, but there is lesion having muscular strength. Such feature of lesion having muscular strength is called BRAWNY, brawny. In case of erysipelas, there is a red skin. It is acute spreading lesion and infected area includes brawny edema with erythema. This is erysipelas. Now, other clinical type of skin infection caused by Streptococcus pyogenes is cellulitis, which is more severe. Cellulitis, it involves deeper subcutaneous tissues. In case of cellulitis, there is involvement of deeper subcutaneous tissues. There will be local inflammation and systemic signs like erysipelas can be seen in case of cellulitis. There is a necessity of surgical debridement also in case of cellulitis because it involves deeper subcutaneous tissues. It spreads very easily, okay, very fast. So we must be very much careful in case of cellulitis. Cellulitis can be caused by various bacteria, but mostly it is caused by Streptococcus pyogenes. So overall there are 4 types of skin infection caused by Streptococcus pyogenes: impetigo, pyoderma, erysipelas, and cellulitis. Regarding whole chapter of Streptococcus pyogenes, you follow the first year. Bacteria Streptococcus pyogenes, which was taught in respiratory system infection. Okay, so in this integumentary system, just a brief concept of skin infections caused by Streptococcus pyogenes. Now we'll jump to other chapter, that is Bacillus anthracis, which is causative agent of anthrax. Have you heard about the disease anthrax? It is caused by bacteria called Bacillus anthracis. We'll study in detail of Bacillus anthracis in today's class. First, morphology of Bacillus anthracis. Bacillus anthracis, it is a gram-positive bacilli. It is gram-positive. It appears purple or violet in color, but it is bacilli, rod-shaped bacteria. It is gram-positive bacilli. It is a spore-bearing bacteria. Bacillus anthracis, it is spore-bearing bacteria. Shape and position of spore is oval and central. There is oval and central spore in case of Bacillus anthracis. Bacillus anthracis, it is capsulated bacteria. It contains capsule. Already we know that capsule can be composed of polysaccharide, polypeptide, or hyaluronic acid. In case of a different bacteria, there may be different composition of capsule. In case of Bacillus anthracis capsule is composed of polypeptide. Just let me inform you, because this may be asked in multiple choice question also. For example, polysaccharide capsule is present in bacteria such as Klebsiella. Polypeptide capsule, it is present in Bacillus anthracis. Hyaluronic acid capsule, it is present in case of Streptococcus group of bacteria. Okay, here as we are studying Bacillus anthracis today, it is capsulated bacteria, and capsule of Bacillus anthracis, it is composed of polypeptide. Similarly, while studying morphology of Bacillus anthracis, we have to add about motility. This bacteria is non-motile. It cannot move because it do not contain flagella. So in morphology of Bacillus anthracis, it is gram-positive bacilli. It is spore-bearing. Shape and position of spore is oval and central. It is capsulated bacteria. Capsule is composed of polypeptide and it is non-motile. Now, cultural characters for Bacillus anthracis. It is aerobic bacteria. It grows best in presence of oxygen. It is aerobic bacteria. It grows best at 37 degrees Celsius. Now, what are different colony characters seen in different culture media in case of Bacillus anthracis? On blood agar, there will be non-hemolytic colonies. In case of Bacillus anthracis, on blood agar, there will be presence of non-hemolytic colonies, no hemolysis, gamma type, gamma hemolytic or non-hemolytic. On nutrient agar, special type of property we can observe. There will be appearance of medusa head colonies, MEDUSA, medusa, HEAD, head. Medusa head colonies will be seen on nutrient agar. Now, what is medusa head? Medusa head means edge of colony which show wavy margin and radiating projections. Wavy margin. Margin भनेको कोलोनीको पेरिफेरीमा if you use the hand lens and if you observe the margin, you can see wavy margin and radiating projections. This type of colony character is called medusa head colony, which is a special feature of Bacillus anthracis on nutrient agar. So on blood agar, it showed non-hemolytic colonies. On nutrient agar, medusa head colonies can be seen. Similarly, in gelatin stab culture, gelatin is semi-solid component. Gelatin media is semi-solid media which we prepare in test tube. If we perform stab culture, stab भनेको media हामीले कसरी इनोकुलेट गर्छौं त्यसरी gelatin inverted fir tree appearance. Fir tree भनेको सल्लोको रुख कस्तो हुन्छ त माथि कोनिकल भएर गएको हुन्छ, तल ब्रोड हुन्छ, माथि कोनिकल हुँदै जान्छ हो. That is fir tree appearance. In case of gelatin stab culture, in case of Bacillus anthracis, there will be appearance of inverted fir tree appearance. Mediumमा inverted fir tree appearance. Bacillus on blood agar it shows non-hemolytic colonies. On nutrient agar it shows medusa head colonies, and in gelatin tree appearance. Similarly, in bio little value. But if we perform biochemical test for Bacillus anthracis, it will show catalase positive. Catalase test positive. If we perform sugar fermentation test with certain sugars, for example, glucose, maltose, they will be fermented with acid production. It is catalase positive. It ferments sugar such as glucose, sucrose, maltose with acid production. Acid production means sugar media originally kasto huncha, pink huncha, but if it gets converted into yellow, it is fermented. Similarly, other biochemical reaction, it reduces nitrate to nitrite. It is catalase positive. Glucose, sucrose, maltose, sugar are fermented with acid production. It reduces nitrate to nitrite. These are biochemical tests shown by Bacillus anthracis, but all these biochemical tests are of little value for identification of Bacillus anthracis. Now, determinants of pathogenicity. Everyone, can you hear me? Response, please.
Yes, sir.
Okay. Now, determinants of pathogenicity. What are various virulence factors in case of Bacillus anthracis? Already we know that it is capsulated bacteria. If bacteria contains capsule, remember one of the virulence factor is capsule for that bacteria. What is the role of capsule? It inhibits phagocytosis. Phagocytosis हुन दिँदैन, ब्याक्टेरियाको सर्भाइभ हुन्छ होस्ट सेलमा disease cause garcha. So one of the virulence factor is capsule in case of Bacillus anthracis. Other virulence factor is toxin, especially exotoxin. As it is gram-positive bacteria, so it releases exotoxin. Exotoxin of Bacillus anthracis, it includes three components: edema factor, protective antigen, and lethal factor. Edema factor, EF, protective antigen, PA, lethal factor, LF. So exotoxin, it includes three components: edema factor, protective antigen, and lethal factor. Mixture of these three components of exotoxin, edema factor, protective antigen, and lethal factor, mixture of these three components collectively is called anthrax toxic complex. Anthrax toxic complex. Okay. Now, pathogenesis, how it leads to anthrax. First, protective antigen, PA, one of the component of exotoxin, protective antigen PA. It binds to receptors on surface of macrophages. Then, if protective antigen, if it binds with edema factor, it will lead to edema, accumulation of fluid. If protective antigen, yes, any question? Do you have any question? Is somebody wants? Okay. So, protective antigen, edema factor, and lethal factor. These three are components. Yes. Bana. स्लाइड देखिरहेको छैन अहिले रिसेन्टली मेरो भिडियोमा प्रब्लम आइरहेछ कि मेरो क्यामेरामा प्रब्लम आइरहेछ ल्यापटपमा अब क्लास मिस गर्नु भएन त्यही भएर अहिले सुन म तिमीहरुलाई यो नोट प्रोवाइड गर्दिउँला बरु है त ल पहिला सुन है सो प्याथोजेनेसिसमा के बुझ त जुन एन्थ्राक्स टक्सिक कम्प्लेक्समा तीनवटा कम्पोनेन्ट छ प्रोटेक्टिभ एन्टिजेन edema factor and lethal factor. Protective antigen, if it binds to edema factor, it will lead to edema. If protective antigen binds with the lethal factor, it will lead to cell death or cell necrosis. So, protective antigen, edema factor, and lethal factor, these are three major components. Protective antigen, it can bind with either of the components, either with edema factor or with the lethal factor. If protective antigen binds with edema factor, it will lead to edema. If protective antigen binds with the lethal factor, it will lead to cell death. Cell death भनेको के त necrosis. So, anthrax in this way, it will lead to appearance of clinical features. Now, infection by Bacillus anthracis, anthrax. Infection by Bacillus anthracis is called anthrax. Clinical condition is anthrax. Disease is anthrax. Anthrax, it can occur in animals and humans. Okay, there may be animal infection with anthrax or there may be human infection with anthrax. First, animal infection. Animal infection. Animals such as sheep, goat, cattle, such animals, they may get infection by ingestion. Okay, animals, they may acquire anthrax by ingestion. If anthrax occurs in animals, animals will die within 2-3 days. Such anthrax-infected animals, before their death, they discharge anthrax bacilli. it means Bacillus anthracis, from their mouth, nose, rectum, etc. So Bacillus anthracis will be released from mouth, nose, or rectum from infected animal which are sporulated in soil. Sporulation भनेको के त spore formation. Bacillus anthracis को spore बन्छ रे, that will be in soil and serve as source of infection. So infected animal हरुबाट soil मा, environment मा Bacillus anthracis को spore formation भयो. These spores of Bacillus anthracis act as source of infection for humans. Okay, now human infection with anthrax, human anthrax. Human may acquire anthrax by either of three routes. Remember, there may be three routes for causing human anthrax. Inoculation. First is inoculation by contact. Next is inhalation by respiratory route. Other is by ingestion. So there are three routes: inoculation, inhalation, and ingestion. 3I. Thinota I. Inoculation, inhalation, and ingestion. Most common route is by inoculation, by contact. So, according to route of infection, either inoculation, inhalation, or ingestion, there are three clinical types of anthrax. If anthrax is transmitted through inoculation or contact, that clinical type of anthrax is called cutaneous anthrax. If anthrax is transmitted by inhalation, respiratory route, that clinical type of anthrax is pulmonary anthrax. And if human anthrax is transmitted by ingestion, it will lead to other clinical type of anthrax, that is intestinal anthrax. There are three routes for transmission: inoculation, inhalation, and ingestion. And accordingly, कुन रूटबाट transmit भयो, on that basis, there are three clinical types of anthrax: cutaneous anthrax, pulmonary anthrax, and intestinal anthrax. Cutaneous anthrax, it is acquired by inoculation. Pulmonary anthrax, it is acquired by inhalation. And intestinal anthrax, it is acquired by ingestion. Is it clear? In case of untreated cases, all three types of anthrax, cutaneous, pulmonary, or intestinal anthrax, all three clinical types of anthrax, if they are untreated, they will lead to septicemia and death. What is septicemia? This is your fourth semester, you know it well. So, all three clinical types of anthrax, if they are untreated, it will lead to septicemia and death. Literal meaning of term anthrax, it is coal, कोइला, C-O-A-L, coal, कोइला जस्तो. Anthrax को meaning हो कोइला, coal कस्तो हुन्छ त कालो हुन्छ. Why this disease is named as anthrax? There are three clinical types of anthrax, among which most common type is cutaneous anthrax. र cutaneous anthrax को feature के हुन्छ त? There will be formation of black color of scar. घाउ सन्चो हुने बेला जुन लिजन माथि बन्ने दाग हुन्छ त्यो कालो हुन्छ. त्यो कालो कलरको देखेर क्युटेनियस एन्थ्राक्समा यो डिजिज को नामै एन्थ्राक्स राखेको, कोइला जस्तो भनेर. Okay, so in this way this disease is named as anthrax according to black color of scars seen in cutaneous anthrax. Most common clinical type among cutaneous, pulmonary, and intestinal anthrax is cutaneous anthrax. Cutaneous anthrax, it is also called Hyde Porter's disease. भरियाहरूको बडीमा स्किन एब्रेजन छ जसले क्यारकास हाइडहरू बोकेको छ उसबाट एन्थ्राक्स ह्युमनमा ट्रान्समिट हुन सक्छ इनोकुलेसन कन्ट्याक्टबाट So this type of anthrax is called cutaneous anthrax. It is most common among hide porters. That's why cutaneous anthrax is also called hide porters disease. Spores of Bacillus anthracis enter the skin through abrasion in face, neck, arms, back, etc. Mostly among farmers and persons handling carcasses and hides. Initially, lesion starts as papule. Papule भनेको के हो त रातो बिमिरा हैन स्किनमा lesion starts as papule then it appears as pustule. Pustule भनेको के vesicle which contains fluid. Cutaneous anthrax मा के हुन्छ त clinical lesion पहिला papule रातो बिमिरा मात्रै देखिन्छ then it appears as pustule. It means vesicle containing fluid. Inflammatory reaction leads to congestion and edema. Inflammatory reaction leads to congestion and edema of affected site with necrosis at the center of lesion. Center of lesion मा चाहिँ necrosis हुन्छ covered by black scar. एउटा लिजन छ होल लिजन माथि ब्ल्याक स्कार बन्ने होइन त्यसको सेन्टरमा मात्रै ब्ल्याक स्कार बन्छ कालो रफ सरफेस बन्छ दाग This central necrotic area is surrounded by vesicle containing fluid. 90% cases of cutaneous anthrax they resolve spontaneously. आफै सन्चो भएर जान्छ 90% मा and only 10% of cutaneous anthrax will lead to generalized infection and septicemia. Cutaneous anthrax, it is also called hide porters disease. भरियाहरूमा छाला बोक्ने भरियाहरूमा एनिमलको छाला बोक्ने भरियाहरूमा cutaneous anthrax commonly देखिन्छ, so it is called hide porters disease. How it is transmitted? If that porter has abrasion in skin, either in face, neck, arms, back, etc. During handling of carcasses and hides, Bacillus anthracis that is present in skin of that animal that will be transmitted to human through skin abrasion in that human being. त्यो भरियामा र के देखिन्छ त lesion starts as papule रातो बिमिरा then it appears as pustule fluid containing vesicle. Inflammatory reaction leads to congestion and edema of affected site with the necrosis at the center of the lesion. Necrosis कहाँ हुन्छ at the center of the lesion covered by black scar. Central necrotic area is surrounded by vesicle containing fluid. 90% cases of cutaneous anthrax they resolve spontaneously आफै सन्चो हुन्छ and only 10% will lead to generalized infection and septicemia. This is about cutaneous anthrax. Now other clinical type of anthrax is pulmonary anthrax. Pulmonary anthrax, it is transmitted by inhalation, respiratory route matter. It is most commonly seen among wool sorters. ऊनको व्यवसाय गर्नेहरू है त. That's why it is also called, that's why pulmonary anthrax is also called wool sorters disease. Cutaneous anthrax, it is also called hide porters disease. Pulmonary anthrax, it is also called wool sorters disease. It occurs due to inhalation of dust containing spores of Bacillus anthracis. Pulmonary anthrax or wool sorters disease, it occurs due to inhalation of dust containing spores of Bacillus anthracis. Bioweapon. Have you heard about bioweapon? Living microorganisms. Just example is Bacillus anthracis that we are studying today. This spores of Bacillus anthracis, they can be used as bioweapon. वारहरूमा bioweapon. यो सेकेन्ड वर्ल्ड वारमा युज भइसकेको हो स्पोर अफ बेसिलस एन्थ्रासिस है त एज बायोवेपन. एक साइडको मिलिट्रीले आर्मीले के गर्छ त स्पोर्स अफ बेसिलस एन्थ्रासिसलाई बायोवेपनको रूपमा युज गरेर अपोनेन्टतिर हान्दिन्छ. सेकेन्ड वर्ल्ड वारमा के भएको थियो त स्पोर्स अफ बेसिलस एन्थ्रासिस दे वेयर युज्ड एज बायोवेपन. बायोवेपनको रूपमा युज भयो. स्पोर अफ बेसिलस एन्थ्रासिस अपोजिट साइडको आर्मीहरूमा गयो. देन दिस bioweapon, it can be spores of Bacillus anthracis, it can be used as bioweapon. This bioweapon, it will lead to death of human beings, which is called pulmonary anthrax, which is by pulmonary anthrax. So there are three clinical types of anthrax: cutaneous, pulmonary, and intestinal anthrax. Bioweapon ले कुन type of anthrax गराउँछ त? Pulmonary anthrax, respiratory infection, र मान्छे मर्छ. It is high risk pathogen, है. Bacillus anthracis, it is high risk pathogen. यो छिर्यो त मार्यो. Okay, and which type of anthrax it will lead to? If it is used as bioweapon, it will lead to pulmonary clinical type of anthrax, pulmonary anthrax. Now next type, one is cutaneous anthrax, other is pulmonary anthrax. Next type is intestinal anthrax. Intestinal anthrax, it is very rare clinical type. Most common is cutaneous anthrax. Then rarely pulmonary anthrax, and very rarely intestinal anthrax. Intestinal anthrax, it occurs due to ingestion of improperly cooked infected meat. कुनै एनिमललाई एन्थ्राक्स लागेको छ र improperly cooked infected meat को ingestion भयो भने it may lead to intestinal anthrax. It is very rare, right? It causes severe enteritis and bloody diarrhea. Intestinal anthrax, it causes severe enteritis and bloody diarrhea. Fatality rate, death rate, it is very high in case of anthrax. So there are three clinical types of anthrax caused by bacteria Bacillus anthracis: cutaneous anthrax, pulmonary anthrax, and intestinal anthrax. Cutaneous anthrax, it is transmitted through inoculation or contact. It is also called Hyde-Porter's disease. Pulmonary anthrax, it is also called Wool-Shorter's disease. It is transmitted through inhalation by respiratory route. Very rare type is intestinal anthrax, which occurs by ingestion. So, three clinical types: cutaneous, pulmonary, and intestinal anthrax. Most common type is cutaneous anthrax. Now, lab diagnosis of Bacillus anthracis. First, we have to collect sample. We can collect swabs, fluid, pus, blood, sputum, etc. We can collect swab, fluid, pus, blood, or sputum according to site of infection before administering antibiotics. Before giving antibiotics, we have to collect various clinical samples according to site of infection for lab diagnosis of anthrax. Then processing. It is high risk pathogen. That's why lab personnel should be protected with additional precautions during handling of specimen. Direct test गरिहाल्ने होइन. Precaution लिएर lab coat, gown, gloves, mask, सबै त्यो स्पेशल प्रिकसन लिने बायोसेफ्टी क्याबिनेटमा मात्रै टेस्ट परफर्म गर्ने इट इज हाई रिस्क प्याथोजेन भनेको व्हिच क्यान बी इजिली ट्रान्समिटेड एन्ड इट मे लिड टु डेथ त्यही भएर ल्याब डायग्नोसिस गर्ने हस्पिटलको ल्याब स्टाफले पनि स्पेशल प्रिकसन लिने अनि बल्ल टेस्ट गर्ने फर ल्याब डायग्नोसिस व्हाट वी क्यान डू वी क्यान परफर्म माइक्रोस्कोपिक एक्जामिनेसन Microscopic examination we can perform gram stain. Already we said that Bacillus anthracis is gram positive bacilli having spore, right? That's why in gram staining what we'll observe if the bacteria is Bacillus anthracis, we'll observe gram positive spore bearing bacilli in chain. Gram positive means purple color. स्पोर बियरिङ भन्ना बित्तिकै ब्याक्टेरियाको सेल भित्र कलरलेस एरिया देखिन्छ कुनै कलर नलिएको दैट इज स्पोर इट इज बेसिलाई ग्राम पोजिटिभ बेसिलाई having central and oval spore. If we perform motility test, this bacteria is non-motile. Already we said it is capsulated bacteria. How capsule can be demonstrated? By India ink method. Negative staining. If we perform India ink staining, we can demonstrate capsule of Bacillus anthracis because it is capsulated bacteria. So microscopic examination, we can perform gram stain, we can perform motility test, we can perform negative staining with the India ink. Similarly, we can perform other tests for Bacillus anthracis that is called Macfadyen's reaction. MC, Mac, F-A-D-Y-E-A-N, Macfadyen. Macfadyen reaction. What is this? When blood film or blood smear is stained with methylene blue stain, we'll prepare blood smear on the glass slide. If it is stained with methylene blue stain, Bacillus anthracis it will appear as large blue bacilli. Bacillus anthracis will appear as large blue bacilli surrounded by a red stained capsule. Capsule periphery ma red color ko huncha re. Bichama Bacillus anthracis bacilli chahi blue color ko huncha re. Polychrome methylene blue le blood smear lai stain garda waripari red capsule ra bichama blue bacilli dekhyo bhane it is Bacillus anthracis. It is called Macfadyen's reaction. Macfadyen's reaction is positive in case of Bacillus anthracis. Similarly, we can perform culture. In culture, how we can identify this is Bacillus anthracis? On blood agar, it will show non-hemolytic colonies. On blood agar, it will show non-hemolytic colonies. On nutrient agar, what head? Medusa head. On nutrient agar, Medusa head colonies will be seen. In gelatin stab culture, what appearance we can see? Inverted powdery appearance. So on blood agar, non-hemolytic colonies. On nutrient agar, Medusa head colonies. In gelatin stab culture, inverted powdery appearance can be seen in cultural characters in case of Bacillus anthracis. Along with microscopic examination, culture, we can perform serological test. In serological test, we can perform detection of antibodies against LF, lethal factor, and EF, edema factor. Lethal factor or edema factor, component of anthrax toxic complex theory virulence factor. We can detect antibodies against lethal factor and edema factor by ELISA by performing serological test. This serological test, detection of antibodies by using ELISA, it is one of the methods for lab diagnosis of Bacillus anthracis. So, for lab diagnosis, we can perform microscopic examination, we can perform culture, or we can perform serological test for lab diagnosis of Bacillus anthracis, which is causative agent of anthrax. So, there are three clinical types of anthrax: cutaneous anthrax, pulmonary anthrax, and intestinal anthrax. According to sites involved, we may collect various clinical specimens and we can perform either microscopy or culture or serology for lab diagnosis of anthrax. This is about Bacillus anthracis and disease anthrax. Now there is one additional topic, very small topic, that is anthracoid bacilli. Anthrax is disease caused by bacteria Bacillus anthracis. Other terminology is anthracoid, ANTHRACOID, anthracoid bacilli. Anthracoid bacilli means Bacillus species other than Bacillus anthracis are called anthracoid bacilli. Genus is Bacillus. It contains various species: Bacillus anthracis, Bacillus cereus, Bacillus subtilis. There are various species of genus Bacillus. Bacillus anthracis, it causes anthrax, but all other species of genus Bacillus, such as Bacillus cereus, Bacillus subtilis, other than Bacillus anthracis, they are called anthracoid bacilli. Anthracoid bacilli, they are opportunistic pathogen. Opportunistic pathogen भनेको के हो त? Normally in immunocompetent people, they cannot cause infection. But if there is immunosuppression, कोही मान्छेमा immunity कम भएको छ भने they can cause infection. त्यसलाई के भन्छ त? Opportunistic pathogen. So anthracoid bacilli, they are opportunistic pathogen. All other features are just opposite to Bacillus anthracis. Anthracoid bacilli को अरु property के हुन्छ त? It is just opposite to Bacillus anthracis. For example, Bacillus anthracis is non-motile, but anthracoid bacilli will be motile. Bacillus anthracis it is capsulated. Anthracoid bacilli will be non-capsulated. Bacillus anthracis, it shows non-hemolytic colonies on blood agar, but anthracoid bacilli, they show hemolytic colonies on blood agar. Bacillus anthracis, it gives McFadden's reaction positive, but anthracoid bacilli, they will be McFadden's reaction negative. So what are features of anthracoid bacilli? They will be motile, they will be non-capsulated, they will show hemolysis on blood agar, they will show McFadden's reaction negative. These are features or properties of anthracoid bacilli. So Bacillus anthracis is high-risk pathogen, but all other species of Bacillus, they are called anthracoid bacilli. They are opportunistic pathogen. They are motile, non-capsulated, hemolytic on blood agar, and McFadden's reaction negative. Example of anthracoid bacilli: Bacillus cereus, Bacillus subtilis, etc. Other than Bacillus anthracis. So they are called anthracoid bacilli. Overall, today we have covered skin infections caused by streptococcal infection. Streptococcal skin infection. Streptococcus pyogenes, it can cause a respiratory infection, skin infection, other pyogenic infection, suppurative infection, and non-suppurative complication, among which all other diseases we have studied in first year. But in integumentary system, today we have focused only on skin infection caused by Streptococcus pyogenes. What are different clinical terms? Skin infections caused by Streptococcus pyogenes: 4. Impetigo, pyoderma, erysipelas, and cellulitis. Now this topic is over. Now next bacteria, Bacillus anthracis. which causes a skin infection. Bacillus anthracis, it causes anthrax. There are three clinical types: cutaneous anthrax, pulmonary anthrax, and intestinal anthrax. Among these three clinical types of anthrax, most common type is cutaneous anthrax. That's why this chapter anthrax is kept under integumentary system. Is it clear? For lab diagnosis, according to sites involved, we will collect various clinical specimens and we can perform microscopic examination, culture, and serological test for lab diagnosis of anthrax. Other terminology we have studied, that is anthracoid bacilli. Bacillus anthracis, it causes anthrax, but all other species of genus Bacillus next to Bacillus anthracis, they are called anthracoid bacilli. They are opportunistic pathogen. They have all properties just opposite to Bacillus anthracis. Examples are Bacillus cereus, Bacillus subtilis, etc. Any query? This is about Bacillus anthracis.